All right, chapter 17 is what finally takes us from looking at DNA and how it's passed down, how it's inherited, what's its structure, right? The genotype of the organism to the phenotype of the organism. And what we're going to find is that proteins are the link between genotype and phenotype, right? So all the information is in the genes, specific sequence of nucleotides, right? A's, T's, G's, C's, all that. And what you've inherited is going to lead to the production of certain proteins, and that is then going to give you these physical traits. So we refer to this process in several different ways. One, the movie talked about this as well. They called it the central dogma of biology, the fact that DNA is what codes for proteins. Sometimes we talk about it as gene expression, right? The process by which DNA directs protein synthesis. And then we'll also more specifically break it down, because gene expression takes two steps: transcription, where we convert DNA to RNA, and translation, where we take RNA and build the corresponding proteins. So if I say any of those things, right, gene expression, the central dogma, transcription, translation, they're all talking about the same processes, and the processes that chapter 17 explains. And these are the processes that are going to start to be able to explain things like everyone wanted to talk about mutations, right? Well, here's an example of a mutation. So this is an albino raccoon, and basically we have a single faulty gene in this raccoon, and that is changing the gene expression, right? And therefore changing the phenotype. So that's what we're looking at. A series of experiments are what helped really scientists figure out that, right, at this point we now know the structure of DNA. Watson and Crick have laid that out. People have started figuring out, right, Chargraph and Morgan and all these people are figuring out, okay, DNA is the genetic material. And then a couple of researchers, Beadle and Tatum, are going to start figuring out, oh, the DNA has the instructions for how to build proteins. And so we won't go into all the details of this, but basically, again, more radioactivity. Biologists just love radioactivities. They were using that actually to stimulate mutations, right? So we were looking for mutations as a way to figure out what the genes were actually doing. And so basically what they ended up coming up with is what was called the one gene, one enzyme hypothesis. So they were making little mutations and seeing little changes in the outcome. We now know that every protein isn't an enzyme, and so now it's the one gene, one protein hypothesis, maybe more correctly stated, the one gene, one polypeptide hypothesis, right? The gene really codes for a string of amino acids, but in order for that polypeptide to be effective, it's got to be a protein. So a lot of times we refer to the products of genes as proteins, even though technically they're probably just polypeptides that then fold on themselves. Okay. So what we're going to do is we're going to look at this on a few different levels, right? And you got to kind of think about, like, how deep do I want to go here, right? So edits from the highest perspective, right, at the base, right, what you need to know, what I would say like any C student has to get down here, right, is that basically in order for this protein production to happen, we're going to go through the processes of transcription and translation. Now, what this image is showing you is how this looks a little bit different between bacterial cells on the left and eukaryotic cells on the right. You should notice that both of them use transcription and translation, right? Eukaryotic cell, there's transcription and translation, okay? But, and we go from DNA to messenger RNA to that polypeptide chain, right, to the protein. We do the same thing in a eukaryotic cell, DNA to messenger RNA to protein. The big difference here is going to be in eukaryotic cells, we have a few extra layers of processing, and we will look at how that works, right? But at your basis, right, you should know that transcription takes the information coded in DNA and uses it to build an RNA molecule, specifically a messenger RNA molecule, or mRNA. That messenger RNA is going to be read by the ribosome, right? The ribosome is an organelle that actually does the translation process. And so translation is taking that messenger RNA code and actually building the polypeptide chain that it instructs. Okay. And we'll come back and we'll talk about that processing a little bit when we see it in eukaryotic cells. So again, well, that just flows through there. So we'll look at this. Okay, but again, that's kind of your like C-level baseline knowledge. You've got to know that coming out of general college biology. Transcription and translation. One of the way I remember this actually, if this helps, right, is so think about DNA is a nucleic acid, right? And RNA is super closely related. It's also a nucleic acid. So when you think about transcription, transcription is just taking notes. Like if you were in the courtroom writing everything down, you're the transcriptionist, right? Translation is when you switch languages. So now we're going from messenger RNA, which is a nucleic acid, to our protein, which is an amino acid, right? So we're switching languages, so we translate from one to the other, if that helps you. Okay, I've already covered this. Moving on, here we go. Okay, so then we're going to step in a little bit closer, right, and see what this actually looks like, right? How are the instructions for assembling amino acids into proteins actually encoded in the DNA? So this is one of the things that threw people off of the chase, deciding whether DNA or protein was the molecule of inheritance, because there's 20 different amino acids, right? And so it was a more complicated alphabet. They thought that was going to be what actually held inheritance, right? So there's 20 different amino acids, but there's only 4 different nucleotides, right, in DNA, A, T, G, and C. So how is it possible that, how do I say this? How do you take the code in that nucleic acid and make the code for an amino acid? And so basically the way this ends up happening, the information from a gene to a protein is based on what we call a triplet code. And so we end up reading that messenger RNA 3 letters at a time, right? A set of 3. Where's my pen? Okay. So again, if this is the DNA, remember DNA is double-stranded. One side is going to be the template. So we're going to go ahead and unzip this DNA, and we're going to use that template strand to direct the production of this messenger RNA. So you'll notice that A brings in a U. We haven't talked about this yet, have we? Remember? Maybe we haven't talked about it. In RNA, we use U, uracil, instead of T, thymine. So anytime that you see an A, now the corresponding nucleic acid is going to be uracil. So if we see an A in the template, we bring a U into the messenger RNA. We see a C in the template, we bring a G into the messenger RNA. And this is that process of transcription. This is actually all taking place in the nucleus, right? The DNA never leaves the nucleus. We're just going to make a copy of it while we're in there. So we've made this messenger RNA. And then when the ribosome goes to do translation, it is essentially going to be reading the messenger RNA 3 letters at a time. Those 3 letters at a time are called a codon. And what we'll see happen is that each codon, each set of 3 nucleic acids, codes for a specific amino acid. So here we see UGG being the code that tells the ribosome tryptophan is going to be the amino acid here, right? Then it moves over and it reads the next 3 letters, UUU, and knows that phenylalanine is the next amino acid that belongs. So what you end up with is you have this genetic code, your DNA, tucked away in the nucleus, right? But the ribosomes know how to interpret that and actually build the proteins that are in the instructions. Now, you're thinking, I don't know how to read DNA, right? How to decipher it. I don't speak that language. So fortunately, some scientists figured this out. And basically what we're going to see, let's go to that one. What we're going to see here is the way that scientists have deciphered this. So let's go back. What was the first? So UGG. So notice what we do here, and we'll do this a little bit in lab in a couple weeks if that helps. So the way you use this table to decipher the code of the DNA, right? This is like a choose your own adventure book or something. What was our code we were going for? UGG. Okay, so if we're looking for UGG, terrible writing. Okay, so what you do is you start out on this left-hand side. It wants to know the first base in your codon, and that was U. Okay, so that's going to tell me that I am in this row, right? I don't have a C, I don't have an A, I don't have a G. So my answer is going to be in this first row. Okay, so then we move on to G. That's terribly written. So now I'm looking up at the top here where it says the second base. G, there's G. Okay, so now I know I'm in the first row and the fourth column. I'm narrowing down my choices here, right? And then our third letter in the codon is another G, okay? And so that codes for tryptophan. They better have chosen that. They did. TRP, the abbreviation for tryptophan. Now, you don't need to memorize all these amino acid names. They're abbreviations. We'll use abbreviations a lot. It doesn't matter if you know what they are. At least not at this level. But some of these you might actually recognize, right? If you shop at Natural Grocer, there's always going to be some sign like leucine, it's the amino acid of the week, right? Here's what it's important for. So you may be familiar with some of these amino acids. And of course, you have to have all 20 of these amino acids in your body, because if the DNA calls for a protein to be made and you're missing tryptophan, you are going to be unable to make that protein. So some of these amino acids are referred to as essential, meaning you can't make them. You need to consume them in your diet in order to have them kind of in that pool available for cells to make proteins. The other ones are non-essential, meaning that you actually have the enzymes to build those specific amino acids, right? And if you think back to, was that chapter 4 or 5 when we were talking about macromolecules, and they showed you that the amino acids are all super similar, they just had the different side chain, right? That's what you're actually constructing. Okay, so a couple other things. I can get rid of that writing for you. Okay, a couple other things to notice about that chart. Of the 64 possible triplets, right? So you have 3 letters. If you rearrange them in every possible way, there's 64 options. 31 of those, 61 of the 64 code for amino acids, right? 3 of them, you'll see up here, are what we would call a stop codon. So that's actually going to be found at the end of the gene. And so we're going to tell the ribosome, okay, you can end the translation here. And then we also have one that can either produce the amino acid methionine, but it also signals it's a start codon. So it's going to be how the ribosome knows to begin building the polypeptide. The other thing that's kind of interesting on here is that this genetic code is kind of redundant, right? So notice like, here's an example right here. UUU and UUC both code for phenylalanine, right? So there's a redundancy here that more than one codon, right, can actually specify a particular amino acid. But it's not ambiguous, right? So no codon specifies for more than one. You could look through here. You are never going to find more than one AAC, right? So AAC always codes for ASN there, right? I don't know what that abbreviation is. Okay. So another thing that comes up then, right, is the ribosome needs to read these correctly, okay? It has to, they call it the reading frame. So it needs to read UGG and then UUU. If it gets off at all, like if it somehow skipped seeing that first one, right, and it shifted where it was reading, we actually would call that a frame shift mutation, where we're looking at the messenger RNA all wrong. We're one letter off, and that's going to change every word, probably every amino acid in this polypeptide chain. So that would be problematic. Okay, So we will practice with this because it does take a little bit to get used to, but realize like this is the level of information or the level of reductionism that scientists have gotten to, that we know what 3 letters in your DNA code are going to tell us what amino acid is going into a protein. And so this ends up being really interesting medically. So there are some disorders, so like cystic fibrosis, for example, mostly we think of it as a respiratory disorder. It affects some other organs as well. But basically there is a single mistake in the genetic code for it to build a protein that transports the ion chloride across the phospholipid bilayer of cells in the lungs and some other organs. And that one amino acid wrong makes it so that this chloride transporter doesn't work. And then those individuals accumulate tons of mucus in their lungs. They get lots of bacterial infections, pneumonia, and typically life expectancy is like 35. Single letter wrong in the DNA changes the polypeptide. So that's the kind of stuff that this ends up being really useful for. And then you can do fun stuff like move these genes around. This is basically showing that all life forms are using the same genetic code. And so what scientists have figured out is you can actually take a gene right here in this, both the pig and the tobacco plant here. We've taken a gene from a firefly. If you ever lived out east, fireflies, they're bioluminescent little bugs. They float around in the sky. They're beautiful. We should have them here. But they do this bioluminescence, this glowing. We can take the gene for how to make that glowing protein, put it in a tobacco plant. Never would have glown. Put it in a pig, right? That one's doing jellyfish. We'll actually do this gene transfer later in the semester, except we're going to take it from a jellyfish and put it into bacteria. No pigs will be harmed in this class, just some bacteria. So, but this helps show that the genetic code is the same, right? It's universal. It's shared from the simplest bacteria all the way up to the most complex animals. And it also shows us that we can move genes, right? And they can still be transcribed and translated, right? So we can produce proteins from other species. Okay, so now we're going to go even further, right? So again, you need to have this big picture that transcription translation occurs. But now and then you can go to the level where you understand this idea of codons and how a ribosome might read it. And now we're going to look even closer. So we're going to dive in here to transcription. So transcription happens in 3 steps: initiation, elongation, and termination. Right? So I guess I can go with this one. So in initiation, right, what's going to happen here is that an enzyme, RNA polymerase, so you might recognize that, right? Polymerase, it's going to make a long strand, but this time it's making RNA. So it attaches to a region on the DNA called the promoter. And we'll talk about that a little more, right? But there's basically a marker on the DNA, and that's how RNA polymerase knows where to attach. Okay? It actually unwinds the DNA, right, and is going to start building. So we don't have a separate, say, like a helicase in this situation. It's actually the RNA polymerase that unwinds, right, and is going to get things started. The next step then is elongation, and this is when we're actually going to start building this messenger RNA, right? And so you can see this coming out. Notice that messenger RNA is also built in the 5' to 3' direction. Okay, so it works the same way. The phosphate is on the 5' end, it's blocked. You have to add to the 3' end. Okay, and so what we're doing then in that case, right, is, does it show us which one? Yep, this is our template strand then, this bottom one. Okay, and so we're reading from 3 to 5 on the template, but we're building messenger RNA in the 5 to 3 direction. Sometimes that confuses students, right? So in this elongation, it's still that RNA polymerase that basically is bringing in the complementary RNA nucleotide. So it brings in, when it sees a T, it brings an A. When it sees an A, it brings a U. When it sees a G, it brings a C. When it sees a C, it brings a G. Hopefully I said that right. Okay. RNA polymerase doesn't need a primer, so that's a little different than we saw in DNA replication. But otherwise, it's actually very similar. Termination then, right, we're going to see basically what's going to happen is we're going to run into a stretch of DNA that signals, okay, that's the end of the gene, because remember, there's all sorts of other stuff out there as well. And so the RNA polymerase comes off, but now we have this RNA transcript. Now, when you see the word transcript, that means that it's preliminary. Other times you might see it called pre-mRNA. This is not the finished RNA product. We still need to do some work. I thought you might, okay, this is even one step further here. So notice what happens here in initiation, right? So in initiation, we said we're looking for a promoter region. That's where RNA polymerase knows to attach, okay? The signal that you're typically going to see is what's referred to as the ta-ta box. It's literally a segment of DNA that has a bunch of A's and T's in it, right? So here you see TA, TA, AAA, right? But so TA, TA is going to be this signal as part of the promoter. Okay. Other things coming in here, what they're calling transcription factors. These are kind of lining up on the DNA to actually help the RNA polymerase when it comes in, that it's able to actually attach properly and do its copying. Okay, so, and we'll see this actually in some of the BioFlex videos. They'll talk about transcription factors, right? It's just these molecules that come in and kind of set things up, right? So they're not super active. It's the polymerase enzyme that's actually catalyzing the reaction, but they're super important in actually making this function properly. We can also kind of zoom in here on the elongation process, right? So when initiation and then elongation, again, emphasizing that it's being built from 5 to 3. And then if we zoom in, oh, there's my marker. If we zoom in, right, we see the template strand of DNA here, and we just see that the enzyme is encouraging that hydrogen bonding between complementary bases. So here's a T. We are bringing in an A. There's an A. We're bringing in a U. Don't forget about the uracil. That's another one that kind of throws people, and understandably so. Okay. This happens really quickly, right? So it's something like transcriptions occurring something like 40 nucleotides per second. I mean, this is like happening really rapidly. And then the other thing to keep in mind is as soon as this RNA polymerase has started elongation and that promoter region is open again, another RNA polymerase could attach on there. And so you can actually have the same gene being transcribed simultaneously, right? Multiple RNA polymerases coming in and making this happen. Because if you decide, hey, I need insulin, you need insulin, right? And so we'll see them pick up the pace pretty rapidly on a lot of these things. Okay, what else? Okay, so that's the process of transcription, right? Again, at kind of multiple different levels there. All right, so the next thing we need to do in a eukaryotic cell anyway is look at this RNA processing. So remember, in bacteria, they don't even have a nucleus. So all of this is happening in the cytoplasm, and basically the ribosome can grab on right away. But here, we are still in the nucleus. Remember, DNA never leaves the nucleus. When you look at something like mitosis or meiosis, the nuclear envelope does fall apart. But other than that, right, it is inside that nuclear envelope. Okay, so in RNA processing, we take that, there you go, pre-mRNA, or what we might call an RNA transcript, okay? And we are going to need to make some adjustments to it. And this frustrates some people, but it's just the way it is, okay? So here's what it looks like. So the 5' end was the first part we built, right? And then we kept building over towards the 3' end. So when the 5' end is done, what we'll do, all of your cells are doing this, right? They add a cap, right? Like a little hat, a cap, onto the 5' end. This does a couple things for us, and notice it's you don't have to go to this level, but it's a guanine, right? We know to the level here that we know it's actually guanine that's added on that 5' end. This is how the ribosome is going to know where to attach, right, and where to start reading. Okay, so that's what the cap is for. The other thing that we'll see happen then is as the pre-mRNA is finished, on that tail end, on the 3' end, we add what's called this poly-A tail, like lots of A's, lots of adenines, hundreds of adenines are added on this 3' end. Pretty much the understanding of what that is for is as soon as the messenger RNA ends up out in the cytoplasm, there's actually enzymes in the cytoplasm that are looking for and breaking down messenger RNA. And again, a lot of times students are like, well, why would you make messenger RNA just to have it attacked? But basically, you might want messenger RNA to make insulin now, but you might not need it an hour from now. So the part of the way this is set up is that it's only the messenger RNA is only active for a certain amount of time. It's being attacked by these enzymes, which will eventually destroy it. If you still need insulin, then you better keep transcribing more copies of messenger RNA, right? So it is useful. Okay, so we see that happening. So part of the processing, we add a cap and we add a tail. One of the other big things that happens is splicing. So part of processing is this splicing. And so what you see here, again, this is the pre-mRNA up top. Notice there are what are called introns. They're showing those in light pink. And then these red portions, they're calling it a coding segment here. It's also called an exon. Okay. What we do in splicing is we remove the introns. We remove portions of messenger RNA that don't actually code for our protein, and it's the spliced together portion that's actually going to give us the correct protein. And again, students get frustrated, and I get it. It's like, well, why did we build it with these introns if that's not part of the code? Like, it just shouldn't be there. But one of the things we do know is that the same gene can code for multiple different proteins if the messenger RNA is spliced differently. So we have something like 20,000 genes, but you have to be able to make like 90,000 different proteins. And so sometimes it can be alternative ways of splicing this, right? And this is how it's done, right? So this is what's called a spliceosome, which is a super creative name, right? So in this spliceosome, basically what's going to happen is you have protein and what are called small RNAs. And so these small RNAs, they're little segments of RNA, and they're basically recognizing areas on this messenger RNA and helping the protein, the spliceosome, this enzyme, figure out kind of where to make the cuts. Right? And so as we're attaching on there, we're going to pull out that light pink region and fuse together our exons, the parts that we actually want to express. And so this is what this would look like. In some proteins, they talk about these different domains, right? So it's almost like you're building multiple different little parts of the protein and then just putting it together. And so there's actually a correlation there, right? Different exons code for different domains of the protein. And so here they're showing you, right, the green and the yellow and the purple are all exons, and these introns are going to get spliced out. And without them, then we can actually build this polypeptide chain in the order it should be in. So that's that step of processing, right? Going from pre-mRNA, adding, and here, look at this picture, it's on here, the cap and the tail, and we've taken out the introns, and now we only have the exons. It is that piece of messenger RNA, right, that actually is going to leave the nucleus. It goes through a nuclear pore, comes out into the cytoplasm where the ribosome can attach. Okay, the ribosome is where translation takes place, and this is maybe the most crazy and kind of fun part of this whole process. So check out what's going on here. This is the basic concept. I love that on the title here. Okay, so notice what happens. A couple things. So the ribosome is actually in two pieces, right? And it's kind of floating around in the cytoplasm. And when it finds that 5' end, the cap on our messenger RNA, those two pieces are able to like fuse together and clamp around the messenger RNA. So to see it, they're trying to show kind of like a cutaway of the ribosome so that you can see inside. So we grabbed that 5' end, and now we're going to pull the messenger RNA through the ribosome, right, like a old VHS tape, and we're going to read it. And the ribosome, again, helps us read 3 letters at a time, okay? So we see a codon here on the messenger RNA. And now what happens is when we have that codon, we need another molecule to help bring the correct amino acid. And that molecule is called tRNA. tRNA stands for transfer RNA. They actually transfer amino acids to the growing polypeptide chain in a ribosome. So the way this works is, notice here on the messenger RNA, sorry, red might not be the best color, I see the codon, this third codon, GGC. Transfer RNA on one end of the molecule is carrying an amino acid, right? This looks like glycine. Okay, that's one of our 20 amino acids. And on the other end, down here, we have the anticodon. Now I drew on top of it and I can't see. There we go. The anticodon there is CCG, right? So it's complementary. It matches, right, or complements the messenger RNA. So again, just like when we saw like the correct nucleotides coming in either transcription or DNA replication, it's not magic, right? Those are the nucleic acids that are actually going to be able to form hydrogen bonds with each other. And so sure enough, the ribosome is just catalyzing this. It's just encouraging this. But the correct transfer RNA molecule will bring the correct amino acid that was coded for. Okay? And so there's a few different ways to kind of look at transfer RNA, right? They're actually pretty short segments of RNA, and each one carries a very specific amino acid on one end, and then you see the anticodon here on the other end. This is typically how your book is going to picture it, but people have actually modeled this in a 3D way. There we go. Now, how the transfer RNA molecule actually ends up with the correct amino acid is another enzyme, right? So that ASE ending is telling us that this is an enzyme, and basically it's going to read the anticodon, right, fit into the enzyme, and then we're going to attach our amino acids on the other end. So we have to have the correct match there, and then this enzyme will help the tRNA hold on to that amino acid, right? You need an actual bond. Oh, there we go. Look at that. They're actually showing, right, that to build this more complex molecule, right, to actually add the amino acid onto the tRNA, it actually takes cellular energy. It takes ATP to form that bond. Okay. Details here, right? And so again, some kind of computer models of how this happens. We're going to zoom in here and look at a little more cartoon kind of version of what this looks like. So inside of the ribosome, you have 3 different sites where these tRNA molecules will fit. Basically what you'll see is, I should draw over here. tRNA molecules are always going to arrive at this A site. A apparently doesn't stand for arrival, but that's what I like to think of, right? It's then going to scoot over to the P site, which is where the polypeptide chain is actually going to be built. And then eventually that same tRNA molecule moves to the E site, or the exit site. So it arrives, the polypeptide chain is built, and then it exits. And this is kind of what it looks like. So this tRNA molecule just came in because its anti-codon matched the codon, right? And we have another amino acid to join to this chain. Okay, so they're zooming in here. They're trying to show you, right, that these two agreed with each other, right? Oh, we don't care about that. Okay, so the way that this protein is actually going to get longer, let's see if this works if we look at this cartoon. So we have our transfer RNA molecule right here with a growing polypeptide chain, right? So it's in the P site. When another transfer molecule arrives at the A site, what we're going to see is now our amino acids are actually pretty close to each other, right? And so that's part of how this works. They're putting them in the proximity where they could actually bond together. Do you remember what the bond between two adjacent amino acids is called? It's a peptide bond. Do you remember how it happened? What did we take out? If you said water, because it's a dehydration synthesis, you are correct. Okay, and so so that's how it works, right? The polypeptide, or sorry, the peptide bond forms between these two, right? And so we see that now this one no longer is holding on. Everything's kind of moved over here, and then the ribosome is going to shift everybody over, right? So that tRNA gets kicked out of the exit site. The growing polypeptide chain is back in the P site, and we're ready for yet another amino acid to join us. Okay, there's a lot going on here, right? But the big picture, I would say is this: the ribosome reads the messenger RNA and helps bring the correct transfer RNA and therefore the correct amino acid and builds a polypeptide chain. Okay, we will walk through this. We'll do like a little simulation in lab in a couple weeks. And so it is good to know where that A site, that P site, and the E site are. That will make your life easier in lab. Okay, but now we're putting this polypeptide chain together. We went from a DNA code in your nucleus to now actually constructing your flesh and your blood, right? Proteins. Okay, and then eventually we will hit, so remember I mentioned there were three different stop codons, right? And so what they end up inserting is not a tRNA molecule, but this release factor. And so now as the tRNAs scoot out, it's done, right? And the ribosome actually comes apart, and it's going to go look for another 5' cap on messenger RNA to grab onto. Now, as complicated as that looked, it's actually much more complicated than this. So what we'll see are if those proteins are bound to go for export, like if they need to leave the cell, typically that synthesis, that ribosome will actually be attached to another portion of the cell called the endoplasmic reticulum. The ER, which is much easier to say than endoplasmic reticulum, the ER is going to help kind of modify proteins. It's often involved, say like you have like a lipoprotein, you might need to add a lipid on there, right? So you can do those modifications and things at the endoplasmic reticulum. Then you would send them to the Golgi apparatus, all these organelles we'll talk about in a couple weeks, and ship them out of the cell. But what's so anyway, one of the really neat things that researchers have found, so this is an actual microscopic image, okay, showing this mRNA strand and ribosomes all the way around along it. And so now we've got a little cartoon picture up top here showing that just like how we said the RNA polymerase, as soon as it's moved away from the promoter, anot' one can come in and make another transcript. The same thing happens with ribosomes. As soon as that 5' end is open, right, here's our 5' end, the cap, here comes another ribosome, right, to attach to it. And so in this way, you can produce a lot of protein really rapidly. So you make lots of messenger RNA copies, and then you have lots of ribosomes working on those same messenger RNAs at the same time. And so they call that a polyribosome, or sometimes you'll see it called a polysome. There you go. Oh, this is taking it one step further. So this one, they're actually showing you've got RNA polymerase. And because bacteria don't have a nucleus, as soon as the messenger RNA starts coming out of the polymerase, ribosomes are there to grab it and start copying. So that's pretty crazy. All right, so here it is in a large summary picture. I'm actually super impressed with how much detail this artist was able to put in here. Again, noticing things like transcription taking place in the nucleus, right? They're even showing you that it's an RNA polymerase molecule, right, that's going to make our RNA transcript, which then gets processed. We see the introns being removed. We see that poly A tail being attached. Here it's snaking out of the nucleus. The ribosome is going to find that 5' cap, right, and start the translation process, right? Again, pointing out codons and anticodons. This would be an amazing picture just to like print out, hang on your refrigerator, right? Study it a little bit every time you walk by. Mutations, right? So at its basis, a mutation is a change in the genetic code, right? Period. Mutations are a change in the genetic code. But these can actually happen a lot of different ways, and they have different levels of impact, right? So what's called a point mutation is when you just have a single nucleotide pair that changes, right? So here we go. In most individuals, your red blood cells should be this nice fluid molecule that can bend and kind of squeeze through capillaries. And then individuals who have a mutation might have a protein called hemoglobin inside that red blood cell that makes them stiff, particularly when they have low blood oxygen. And so it turns into this kind of sickle shape. So notice the genetic code here, right? They're pointing out that this sickle cell, and we saw this in the movie, right? That young man who had sickle cell disease, this is a point mutation, right? We changed this base pair to this base pair. And right, you're like, well, they're just flipped. Yep, that is enough to make this code wrong. Because look what happens in the messenger RNA. Instead of reading GAG, which if we went to our little table, we would find codes here. Instead, now we read GUG, right, and we get valine. So, a single base can change a single amino acid, and that can lead to really big problems, right? And so sickle cell anemia, super painful. Yeah, we saw a little bit about that in the video. We can talk more about sickle cell. Okay, there's some other mutations that can take place. And so they're giving you a few different examples here, right? So for example, let's see, how do they show? So a silent mutation would be basically a point mutation, but one that doesn't change the amino acid, right? So they have no effect, silent mutations have no effect on the amino acid produced, because remember, sometimes in that code, there were multiple codons that would give you the same amino acid. And so that is a silent mutation. Like, you just got lucky, right? Sometimes there are what's called a missense mutation, right? And so this would be, it still codes for an amino acid, but not the correct one, right? And so that would be like what we just saw in the sickle cell disease. That would be an example of a missense mutation, because now we're going to make the wrong amino acid. Sometimes you can have, again, just a single, like a point mutation, or what they're calling a nucleotide pair substitution. That it's a point mutation. It's one problem. Sometimes it's what we call a nonsense mutation. So the change in that amino acid codon, it gets turned into a stop codon, right? And so you're going to cut off the polypeptide chain early. So most nonsense mutations lead to a non-functional protein. Okay, there's also some kind of bigger problems. And so this is going to be, they're pointing out here, we're inserting, right, another pair, or deleting a pair. And so this can run into problems of reading, right? And so these can be any of these mutations, I suppose, can be bad, but these typically cause bigger problems. So notice here, this is an example. This is called a frame shift. So in this example, basically what we've done is we've added an extra, they're showing an A here, right, and an extra T. And so what happens in the actual messenger RNA code is we end up with an extra nucleotide, and so we shift the frame. We're now going to read our codons still 3 letters at a time, but we got off a little bit, and now everything is going to be wrong from here on out, right? Here's another frame shift mutation that's a result of a deletion, right? So if we took a letter out, everything gets scooted over, right? And again, we'll make the wrong amino acids from then out. And what's this one? A 3 nucleotide pair deletion. Oh, well, that's interesting. So you could just be missing basically an entire codon, and so then you're missing an amino acid. And again, it's wild to think, but a single amino acid missing can mess up that entire protein. Okay, so let's we'll definitely talk a little bit more about transcription translation. We'll watch the video, see if we can answer questions there. We'll spend a little time maybe talking about mutations. I would love for us to think about how these occur maybe a little bit more, and then let's talk about sickle cell a bit more as well. Someone remind me if I forget to talk about sickle cell disease, because that's actually a really interesting link between what we see here in transcription translation and also how it's inherited. So we can go through that. Okay, have a good one.